What's up, everybody? Welcome to Logical, Plausible, Probable, kind of a impromptu, quasi-impromptu live stream going to do here with Sal Cordova in preparation for his upcoming debate tomorrow night with Dr. Dan Cardinal over on Modern Day Debate. And we're going to be actually discussing today some of the interesting aspects of protein improbabilities, the quaternary structure components, and how that creates entirely new, or not new, but additional layers to the improbabilities, both from just a pure mathematical perspective, as well as implausibility in relation to, do you actually think this is a reasonable conclusion? And then we're going to talk about this in relation to application of in abiogenesis and just evolutionary theory in general. So, Sal, thanks so much for taking the time to do this and share your knowledge and research and insight on this very interesting topic.
Thank you.
Now, before we get started, you got your debate tomorrow night. Are you excited about having the backlash response? I think is the title they've got over on MDD. How are you feeling about it?
I'm feeling really good. I think, first off, there's Dan and I have had a history on the internet as not being friendly, but I think we've been able to find a way to be civil with each other and talk in scientific terms. So I want to express my gratitude to him because he actually, on account of him, he actually brought me out of obscurity. So this is one of those things.
And so it's very interesting how, especially from the keyboard warriors, how when a lot of them talk a very big game, but once you actually have to have a face-to-face, or in this context, live video to live video, confrontation or dialogue, suddenly people return to not all, but many return to some semblance of humanity in their conversations. But
I'm excited because a lot of the a lot of the personal stuff is set aside and we're just going to talk science. And I think actually that's why the case for creationism, even though it's going to be primarily about genetic entropy, but then creationism is kind of always in the background. And so I'm really excited. One reason I'm especially excited is if we had this debate 10 years ago, I'd probably be saying, I don't know, I don't have evidence for this or that. But that's totally changed now. There'll be papers I'm citing that are just like released a month ago. And I was just like, there are people in the chat. I think his name was Stupid Horror Energy. And
are you talking about from y'all's first debate?
Yeah.
Oh yeah.
Well, if I'm misidentifying you, I apologize, Stupid Horror. But
hey, all I gotta say is if you willingly pick that as your username, you have also willfully accepted any kind of mocking and derision that may also come your way.
So I thought he or she said something about transposons, and that's all changing real fast. I mean, like I've seen papers and bioarchive stuff coming out like in the last month, so that's why I'm especially excited.
Well, it's interesting you talk about the transposons. Just the other day, Standing for Truth and myself, in the debate we had with Team Skeptic and J.L. Warren. I mean, they were trying to use transposons, ERVs, etc., as just these inarguable proofs of their position and just decimation of ours. And it's, we called them out on it multiple times. It's like, you do realize that there's papers, kind of what you're referring to, right? Like that are coming out. They've been coming out prior to this, but there's also ones coming out, as you just stated, within the last month and a half that are directly contradicting these traditional talking points.
So, it's the biophysicists, it's the structural biologists, the biochemists that are coming out and doing and really winning the debate. I'm just a reporter and the messenger, but they're still, people are still quoting obsolete knowledge and evolutionary literature. It's all, the only people are who are maintaining that view are the evolutionary biologists and stupid whore energy. I mean, that's just, it's not science. It's not biophysics. It's not biochemistry. It's not structural biology. That's about, so that's why I laugh. That's why I laughed at that username. I said, that's almost really appropriate for the level of knowledge that's being represented for someone defending the idea that the transposons are junk, because they're very essential to the structural configuration. And there's Dan Grauer was saying, oh, look at all those random, that's just junk RNA coming out. It turns out those are very important for structural properties of the 4D nucleome. And that's why I'm excited, because I have nothing personal against Professor Stern, but this is going to be an argument on the latest data, the research experiment, and so the the evidence is on our side, and God has made it available to us in the 21st century. Here in the year 2020, this debate probably would not. I probably wouldn't have a chance against Professor Stern Cardinal if this debate took place 40 years ago. But it's a new ball game now because of all the advancement in structural biology and biophysics. So yeah, I'm totally stoked for tomorrow. So I think I'd like to let me see if I can get my screen share here. And John, are you still there? Oh, all right. I think I'm still live. I could see it on my other monitor. So I'm going to try to bring up a screen share here. Let's see if I could do this. So I'm bringing up the screen share, and I hope John's able to reconnect. Hey, nice to see you.
I'm back. Yeah, I'm not sure what just happened.
You got cloned, buddy.
I saw that. Okay, you ready to share your screen?
Yep, I'm going to share audio, so I may have to cut my mic, or either of us. But first, I'd like to share something from Dr. Sanford. Some people are aware of this, but we can all be in prayer for him. He has a heart and lung condition, and there are a lot of people that love him and care for him, and would really miss him if he's gone. So I'd really like to salute him here. I have a 2-minute video from him first. And let me see. I'm going to do it again. I'm sorry. I didn't click the share audio. I'm sorry, John.
No worries.
Okay, try it again. So this should. Alrighty. If I could, if we could give a special tribute to the SFT channel that helped me get all these debates and exposure, and help me stop being invisible. SFT. So
what's SFT?
Standing for truth.
Okay.
Standing for truth.
Hey Sal, something just happened to the video.
If I could, if we could give a special tribute to the SFT channel that helped me get all these debates and exposure, and help me stop being invisible. SFT. So
what's SFT?
Standing for truth.
Okay.
Standing for truth. So they got me my start.
Okay.
So thanks.
Okay. Well, that's great.
Okay.
All right. So I'm John Sanford, and I've been a geneticist throughout my career, and I am a Christian, and I've been basically for the last 20 years, I've been using my science to defend Christian faith and to show that the Bible is credible and that it is, in fact, the best explanation for in terms of origins and in terms of endings, how, from beginning to end. So as I've done this research, it's been my privilege to interact with a lot of very diverse people. And so I would just like to say thank you to Sal Cordova for the collaboration we've had. So Salvador, basically, I contracted with him to do independent research on, especially in the area of biological design and genetic information. And so Salvador has been a great encouragement to me, and he's doing really good work. The Nylanase paper that we produced together, I think is very significant and shows that genes and proteins cannot arise spontaneously. And so that's just a really fascinating topic. And so I just I'm really excited with where Sal and I are going with this. We know that life is designed, and we can show very compelling evidence for that. And the evidence is pouring in now from all directions. So we have now the challenge of sorting out the best information to encourage others. And so that's our relationship, and we're hoping to do videos and books and articles, and I'm excited about my work with Salvador.
If I
That's awesome, Sal. It was a very nice endorsement of everything that's getting excitement about what's going on, and I know you're going to be sharing some of that, some of those discoveries with us right now. Sal, I think you got your mic on mute.
Okay, I unmuted it. So yes, so John and I had been working for 6 years, and maybe only 30 people on the planet knew about my work. And I said, John, we need to try to reach out. And both of us agreed that maybe making videos and outreach like this would be really good, so more people get to hear it. So I really want to express my thanks to the SFT channel and to you, Logical, Plausible, Probable, for helping us get exposure for the work that has been done for the last 6 years and has been pretty much invisible. So today I'm going to be talking about understanding protein probabilities and why abiogenesis and evolution needs miracles. I have no idea how long this is going to take.
We got time. Go for it, man.
One of my favorite quotes from James Tour: "Origin of life research is retarded." And there's a picture from Dumb and Dumber. And so origin of life research in a nutshell. You're telling me there's a chance. So I'm going to give a video from Dumb and Dumber, if you'll permit me.
Mary? I desperately want to make love to a schoolboy.
Maybe I should get going.
No. No, that's not what I meant. What I meant was, I like you, Mary. I like you a lot. I want to ask you a question, straight out, flat out. I want you to give me the honest answer. What do you think the chances are of a guy like you and a girl like me ending up together?
Well, Lloyd, that's difficult to say, and we really don't...
Hit me with it. Just give it to me straight. I came a long way just to see you, Mary. Just, least you can do is level with me. What are my chances?
Not good.
You mean not good like 1 out of 100?
I'd say more like 1 out of a million.
So you're telling me there's a chance? Yeah! Mary?
All right, so I'm sorry that the videos were kind of quiet. So that's something to work on. I don't know why it doesn't come across so loud on the StreamYard. So that's something that can be worked on. So basically, Jim Carrey's excited that she's when the girl said his chances aren't good, and he said, "So there's a chance." So that's how I view Origin of Life research, and that's why I think it's retarded. And anyway, a little bit about my background. Many of you know that I was an engineer in the aerospace industry, but I got trained also in biology so I could work with John Sanford at the NIH. This used to be private knowledge, but it's now public knowledge. Pictured here is Eugene Koonin. He's one of the top evolutionary biologists in the world, and he works at the NIH. And one of his staff members is right here. That staff member is Igor Rogozin, and I'll actually be quoting some of his work. Igor Rogozin was my professor of bioinformatics at the grad level, and he also taught me a little bit of evolutionary biology. So that's just a little bit of background. I'm going to be sharing some of this. I'd also like to introduce Dr. Joseph DeWeese. He is a young Earth creationist, and here he is at a in a prestigious scientific journal, Nature, on the topoisomerase enzyme. Now, I'm really sorry that we're having some audio issues playing it back in StreamYard. I was going to play his testimony. It's only 6 minutes long, but we'll forego that for today until we can figure out the technical difficulties. He's a Christian. He started studying biochemistry because his brother contracted cancer, and he started wanting to understand what cancer was, and so he started studying biology, and it led to a study of biochemistry. And he is a young Earth creationist. I had done some work with Dr. DeWeese, and this is from the this is a abstract from title of an abstract from the FASEB journal. It was for a poster we presented at the Conference of Experimental Biology in 2019 in Orlando. So I will cover some of that. So I've had the privilege of having Dr. DeWeese as a lead author and me as a co-author, and I will talk about protein probabilities from the perspective that I've learned from him. Oh, Salvador Cordova, speaker, closer to the mic. Okay. Is that better? Can you hear me better now?
Yeah, we can hear you fine. They were talking about in context of the video, I believe.
Oh, okay. So let's just talk about some protein probabilities. First thing I'll say, a lot of creationists keep quoting acts or numbers for protein probabilities. That's not really, I would discourage that because every protein is different, and it also means it has different probabilities. The probability of making a paperweight by a tornado going through a junkyard is very different than making a 747 or any complex machine. So there's some proteins that are only 50 amino acids long, and then there are others that are 30,000. And so there are different probabilities for each protein. So I'm not trying to say Doug Axe is right or wrong. I'm just saying that a probability we calculate for one protein can't be generalized to another. So that's just something I want you to have in mind. So let's just start with collagen because that's an easy one to understand. So we'll work with collagen. Collagen you'll find like at Walgreens. Here's a beauty product that's based on collagen. It's trying to make you more beautiful, but collagen is a real protein, and about 30%, 20 to 30% of our bodies are made of collagen. So it's very important protein. This is the spelling of a collagen protein, the collagen type 1 alpha 1 paralog. And I'm going to show you. Now, can you read that? There's a subtle structure here, and I'm going to highlight it. Okay. Do you see it now?
Yep.
Yeah. Yeah, that's a non-random structure. That's very important for the structure of collagen. That's why it can, let me just back up a little bit. That's why collagen is able to make these fibers like that. So that alternating pattern is really important for making it. And so you could see it right there. The G stands for glycine. So now I'm going to show you something a little bit about phylogenetic reasoning. So here's the collagen in a human being, and here's the collagen in a zebrafish, and you could see they still have the same kind of architecture with that alternating, with that glycine every third position. So there's about 75% similarity depending on the scoring technique. And that's why evolutionary biologists will say, oh, that's proof of common ancestry. Okay, it's more subtle than that. Right, and I'm going to cover that, and then I'm also going to cover protein probabilities. So please stop me, John, if there are any things that I...
No, it's making perfect sense so far.
Okay, great. One thing I'll point out is appearances can be deceiving. For example, this pencil's really not bent, is it? It's just the optical illusion of the water and the air. It's something called Snell's law. Also, for thousands of years, people thought the sun and stars orbited the Earth, but we now know that the Earth orbits the sun, and that the Earth is spinning. So things that look superficially one way turn out to be the total opposite the other way. So the reason I'm pointing that out, evolutionary biology is the same way. Superficially, there are times if you look at limited data sets, it'll look like common descent. But when you look at the complete picture, you see that it's a ridiculous conclusion. Or at the very least, common descent would need miracles to make it happen. Common descent needs miracles to make it happen. And that's the thing, is evolutionary biologists do statistical phylogenies like this. They'll compare humans and zebrafish and build all these little phylogenetic diagrams here. In fact, I learned how to do that. It's pretty easy. You just take these protein sequences and then you put it through a phylogeny program like Mega. It's not hard at all. And it's like, well, yeah, look at it. It creates this nice nested hierarchy. They say, well, the evolutionary conclusion is obvious. And it's like, well, that's just, looks can be deceiving, just as in this case. So the evolutionary biologists have said, well, why would God make it look that way? And I'd counter by saying, well, why did God make it look like pencil bent? Is God being deceptive because the pencil looks bent? Or is God being deceptive that it looks like the sun rises and sets every day, and it looks like the stars and rise and set every day? And same with the moon, when it really is that the Earth, it's the Earth spinning that's causing that apparent motion. It's like, no, God's not being deceptive. It may look that way, but if you just look a little more carefully, you'll see the truth. And that's the problem, is when we look at limited data sets, we get the wrong conclusion. And that's what I'm going to argue a little bit today. That's still not protein probabilities. We'll cover that, but I needed to point that out. So Stephen Gould said, did he create to mimic evolution and test our faith thereby? And I'll say, absolutely not. Not any more than God made pencils look bent in water, or he made the world look, he made the universe look a little bit geocentric. That's not being deceptive, because he'll give you the data that you could figure out the true answer. And we have the data, unless you're willing to be willfully blind to it. So I'm I'm not going to belabor this. If you want to talk about Snell's law and also geocentrism, we can talk about that. Hopefully a lot of you know this. So I showed the collagen protein. Now look at this one. This one is the human zinc finger 136. Do you see the pattern? I'll show it to you. Isn't that cool? So you could see that the pattern is different than collagen. It's a different pattern. There's no way, by the way, I've asked evolutionary biologists, can you tell me what the common ancestor of the collagen protein and the zinc finger protein, tell me what it looks like. How does random mutation make one of these from a common ancestor? A lot of them will say, no, we don't believe there's a common ancestor for this. I said, yeah, that's exactly the point. There's no common ancestor for all proteins. And you know that, and you're not telling people that. So we can kind of do the same thing. Oh, let me just give a few more fine points to the zinc finger protein. So each of these letters stand for an amino acid, and there's the amino acid table there. And so C stands for cysteine and H for histidine, and it has all these properties that are represented by this Venn diagram. Now, let me add a little more coloring. We can actually see that we can actually even group, like say this, let me see. Yes, so like your Fs and your Ys are in one group, the aromatic groups, and then this one, the IFL, these are, I don't know what this one is. Speed of Sound should know this one. I'm kind of embarrassed. Anyway, it's in this big orange group here. So you can actually group it, and you can see the structure starts to emerge itself really well. So the point is, we could take these. We can take, say, the human zinc finger 136 and the pig zinc finger 136. They look very similar, about 50 to 60% sequence similarity. We can actually make more of these phylogenetic trees, and it looks like, well, that looks like so much like common descent. But the problem is, again, like with geocentrism and bent pencils, it's only superficial, because you could see the architecture here. On the left is collagen, on the right is the zinc finger 136. So even within the family, it looks like you have evolution. But across families, this is an orchard, basically. So in creationism, you hear the word orchard. Do we have, is it a universal tree, or is it an orchard? Well, in protein, it's so clear. And when I present this to evolutionary biologists, I say, tell me what the common ancestral sequence to both of these looks like. They can't. Easy time saying there's a common ancestor to all of the collagens, and there's a common ancestor to all of the zinc finger 136s. Then when it comes to unifying both of them, they're drawing blanks. And this is kind of the problem of orphan genes or taxonomically restricted genes. It's that way all the way down. And so one way that I could picture this, so if we look at these as either devices, machines, or machine parts, we can look at this. So pianos will have a certain architecture, but you can build them slightly different, just like you might have a pig and a human protein slightly different, but they obey the same architecture. Same for cars. You have cars of different architectures and blenders. But these are distinct architectures. They don't really even conceptually from the same device. It just doesn't make sense. And that's the issue with proteins. So even though we can make trees for the collagen and we can make trees for the zinc fingers, it's a force fit to give them one universal common ancestor, and that universal common ancestry at the protein family level, it's just gone. If you doubt me, ask I invite you to ask dozens of biologists, do you think all major protein families share a common ancestor? Do you think there's one sequence, one gene where all proteins descended from by a process of mutation and natural selection? And most of them will be like, nah. Okay, so common ancestry is broken right there, at least at the protein level. It's very defensible. So that's the orchard model for proteins. So this was actually a picture for organisms, but it works really well for proteins. So that's getting into protein probabilities. Any questions so far? Let me look at the chat. Okay.
So it seems to me that what you're
All
kind of
right.
just displaying so far, Sal, is that yes, there are similarities. However, they don't have they have similar architecture, but not the underlying requirements for ancestry based on the phylogeny methods that are leveraged by evolutionists. Is that correct?
That's correct. Now, I'll tell you why. So, because I know how to do some of the statistical phylogeny, I could take a human, I could take a pig, zinc finger 136, and any other creature, just pull them together in a large data file. I'm just going to put it through a mega computer or phylip or whatever. There are all sorts of phylogeny programs, and it's going to build you this nice little tree. So superficially, that looks like common descent. Okay, and even I would say that superficially. But the problem is, let me go forward. What if I start throwing collagen and zinc fingers in there? Your phylogeny is going to blow apart. If we start, you see, this is cherry picking.
So this
What
this
you
kind
need
of
to do is you just need to take all the genes, throw it in one of these things. And I said, what do you think is going to happen? And everyone said it's going to blow apart. I said, yeah, that's exactly the point. You don't have any hidden Markov models or any of these alignment algorithms that can handle it, because there is no common ancestry among the protein families. That's a known fact. I have some papers. I don't have them handy right now, but I was just laughing. I said, gee, it took you guys that long to figure this out. You know?
Well, it seems to me that what you're talking about also on the reciprocal side applies when you say, oh, they look at, oh, what was this one gene mutation or this one knockout experiment, but then, oh, nothing happened. But then when they knock out two, then there is a massive cascade effect that you're kind of applying the same logic on the phylogeny of proteins of, hey, you look at it only in comparison to one aspect, fine. But then the second you throw in the second, third, fourth variables, then your entire model explodes.
Yeah. This is, I mean, again, what I'm trying to show here is even in the man-made world, we have distinct architectures. A piano is not a car. It's not a blender. But within the concept of piano, you can have slightly different architectures. And so like when there's an architecture of the zinc finger 136 for humans and the zinc finger 136 for pigs. And then we could do the same for collagen. Let me go back here. Well, in this case, it would be the zebrafish. See if I have it here. So we have the collagen architecture, the collagen 1 architecture. There are actually like 30 different types of collagen in each of those. There are many families of collagen, but there's one architecture for the human. Collagen has its architecture and the fish has its architecture. So there is similarity. I'm not going to deny that, but it's when you look at the larger data set, just like what happened with heliocentrism and this Snell's law. When you look at the total data set, you end up like for heliocentrism, they needed to have epicycles to fix all the problems. So when we look at larger data sets and we stop cherry picking, the problems start to emerge for common descent. Okay, we could still, I mean, God bless Michael B. He believes, he still nominally accepts common descent, and I understand that. But he at least knows there has to be, he uses the word intelligent design, but I'll use the word miracle. He knows there has to be miracles to make common descent work. And what I would say is, if common descent needs miracles to make it happen, how is that different from creationism? So
Agreed.
I call this the protein universal common ancestor. I said, is there a protein universal common ancestor? And I use the acronym PUCA, PUCA. PUCA doesn't exist. No common ancestor. So why would God make all these other creatures? Because you'd think it's deceptive. Well, what if God only made humans and no other creatures? Then we'd falsify. We wouldn't have a problem common descent. But how would we have to do biology? Well, we would have to sacrifice each other. We'd have to volunteer human beings for dissections like this one that was done on a fetal pig. And we can go there later. But I think the patterns of similarity and diversity, the patterns of similarity can be interpreted philosophically as a gift from the designer for scientific discoverability, like fine-tuning of the universe. That's just my personal opinion, but I'm finding it hard to run away that all these creatures in biology make excellent model organisms. Oh, by the way, yeah, see, this is a pangenome diagram. Granted, this will be somewhat controversial because it's a little dated, but you could see that like we share some genes with chickens that we don't share with mice. There are some problems with outside, but anyway, we have this thing of model organisms. So when we want to study, how our nervous, our brains work, we study roundworms, and we want to learn a little bit about our nervous system. We study squids. We want to study introns. We can also study plants. And my professor here said, paradoxically, humans share many more introns with the plant Arabidopsis thaliana than with the fly or nematode, which are animals. So these are interesting patterns, but we can study plants. We can blow apart and dissect a plant without having to blow apart a human being, which is great. And we could study chromatin. A lot of the ENCODE work was actually, before that, what began to pioneer it was the study of yeast. And we have other model organisms like bacteria and mice and chimpanzees. So these are gifts from God. That's why we're similar to these other creatures, in my opinion. That's just a theological belief, so that we can understand how we are fearfully and wonderfully made. So rather than looking at the similarity as something that's an irritation to our creationist theory, we can embrace it as something, as a gift from God to help us understand that we're fearfully and wonderfully made. Because the alternative is we would have to volunteer human beings for these sort of grisly dissections. And when I see this, you thank God every day that we're similar to pigs and chimpanzees and mice. That changed my perspective when I saw some of the NIH. So any questions before we get heavy into the protein stuff?
I haven't really seen too much yet, and we're getting ready to go into the real meat here. But I find what you're talking about in relation to we've got these genes in this type of organism and these in the other. But the other one doesn't have this particular one. It's not that they both have it. It's that this type of organism has this gene set, and this other type of organism has a different gene set, but they don't have the crossover. And in terms of what humans have, I find that to be very interesting. And I think it coincides with what you're talking about in terms of the overall structure and adding additional variables to the equation. It really starts to damage, at a minimum, the foundational premise that's being presented as fact by the evolutionary advocates.
Yep. So I'd like to thank all the our faithful listeners that have suffered through the first 30 minutes of me just blabbing about stuff that's outside of protein structure. But actually, just in my defense, we actually did see the first part of protein structure. When we saw the spelling here, I'm sorry to just be blasting through the... So when we have a spelling of a collagen protein here, that spelling, that's called the primary structure. The primary structure is the sequence. So we could say along one dimension, the primary structure, it's kind of a structure if you look at it in one dimension. So the spelling of the protein is the primary structure. So we actually just sort of learned an idea what primary structure is. And in this diagram, it's called the polypeptide chain, but you could say it's the spelling of the protein. That's the primary structure. Now we have things called the secondary structure, the tertiary, and the quaternary, and I'll cover those. But so see, this is nice. You didn't have to go through suffer through 3 years of biology and biochemistry and organic chemistry and general chemistry. You can understand the idea of spelling. I mean, that's a great curriculum if you're going to be a medical doctor or a pharmacist or organic chemist. But for understanding intelligent design and creationism, we could take a few shortcuts to kind of see kind of like the overview, kind of at a high level without getting too bogged down in the details. So the primary structure is basically the protein spelling. So so you can go impress the girls at the bar and talk about protein primary structure, salad bar that is. Anyway, so what is secondary structure? So we have secondary structure. You'll see diagrams like this on the protein, how it coils. Parts of the protein have little coils like this or things that look like sheets. And I'm going to cover secondary structure briefly. So when you spell certain, depending on how a certain segment of a protein is spelled, it may end up forming a little coil that we call a alpha helice. And actually, there's a different kind of helice, and I'm sorry, I'm going to have to just blast through this just so you could see it. In the collagen protein, it actually creates a helix. I don't have a picture of a collagen helix, but you see this alternating G pattern here, the G every third position. That will actually create a helix. It's not an alpha helix, it's a collagen helix, but it looks very similar to this ribbon. So if you've seen ribbons like they often put on decorative balloons, that's what the collagen helix looks like a little bit. And it's also what the alpha helix looks like. So that's an alpha helix. Now I'll show you how an alpha helix is spelled. There are many ways to spell an alpha helix, just like there are many ways to spell a noun. So it's just kind of a general architecture. And here's an example of how we could spell it. So the primary structure is just the spelling in terms of amino acids. And then it makes a alpha. That spelling results in an alpha helix. So why did, just as an aside, why did Urey Miller as an experiment fail? Homochirality is needed for alpha helices and beta sheets. This alpha helix here will break down if you have right-handed amino acids in the mix. It either has to be, to have an alpha helix, it either has to be all right or all left. Otherwise, it breaks down. Urey Miller experiment didn't make homochiral amino acids. And again, origin of life research is retarded. Sorry, I just had to take a little dig there. Because, I'll tell you a really sad story. I'm sorry I misspelled this. Someone I knew in my creationist group, he was an engineer. When he was in high school, someone gave him his teacher gave him the Urey Miller experiment that made him an atheist. It made the student, my friend Ragnar, his name, he was in the Scandinavian countries. So his teacher taught him that, the Urey Miller experiment, and Ragnar became an atheist. As he said, his life went downhill because of his atheism. He found the Lord, thank God, and his life came back. But a similar story for Lee Strobel. He heard of the Urey Miller experiment, and he thought that was proof that life spontaneously arises. And that's the first problem. I mean, if you don't have homochirality, your alpha helices fail, and you're going to have a disaster with your proteins. So we've covered primary and secondary. So you have these little alpha helices. So these are just kind of parts of the protein. The tertiary structure is basically the whole single protein itself. The quaternary structure happens when you have two or more proteins together. Now, I'm not being exactly rigorous with my definitions here, but I'm just trying to give you an understanding of what this is. So this is
this is one of those examples of from the scientific notation perspective, there may be 5, 6, 7, 8, 9 sub indexes of what quaternary can be, and you're kind of giving us the overarching concept. Is that right?
I'm giving overarching concepts, and maybe the way to simplify this, just think of an individual protein. The proper term is polypeptide chain. And when you take multiple polypeptide chains and bring them together, like the helicase or the topoisomerase, which we covered the helicase, you basically have multiple copies if it's homomeric. And I'm sorry to throw out all the terms so quickly, but I just kind of throw out some terms, and we can go back if there's some confusion. It might be more helpful to just keep going through examples. So going back to collagen, I want to show some properties, how some aspects of the collagen. There's more complexity to it than meets the eye. We have something in the collagen known as a localization signal, and that actually tells the cell where to put the collagen for more processing, how to pass it on. This one, let me show a picture of a cell here. So that this localization signal tells it to go to the endoplasmic reticulum. So wherever this thing is, when the gene is nicely spliced and you have the mRNA, there's a localization signal. Actually, I'm not sure. I have to double check this, but I know this is the localization signal. This is telling it, this signal tells where to put the collagen for further processing, and it's going to put it here in the endoplasmic reticulum. So these are like postal codes. So we have zip codes in the man-made world. We have localization signals in the cellular world. So far, so good. So this, I'm just trying to show there's a little bit more complexity. I'm trying to show the meaning of these spellings here.
So quick interjection. So the one of the things I'm deriving from all this is in the context of the evolutionary argument, they might be able to say, oh, well, it can be modified and it still forms a functional insert protein or polypeptide chain name. But it while the code that you're referring to right now might not necessarily forbid the structural aspect of it, it would negate the actual application of that protein in the correct location. Is that or for further processing? Is that a fair assumption?
That's a very fair assumption. What if we didn't have these localization signals in the proteins? So there has to be a policy within the cell, just like there's a postal system or basically internet and routers. What happens if the internet signals are going the wrong way and your little information packets are being sent all over the place? It's going to be a disaster. So so some of these origin of life things, they'll say, oh, we had 13 amino acids. We were able to get catalytic ability. It's like, yeah, I grant that. But that's kind of ridiculous. This is like a tornado passing through a junkyard and making a paperweight. All right. It's easy to make paperweights. It's really easy. But that doesn't generalize something like a collagen protein, or I can even give more complex ones. So
one more question while I'm thinking about, I don't want to forget this. Are these things that you're referring to now, these destination codes, are they being accounted for in phylogeny?
Yes and no. And there's something really interesting. Oh my goodness, we're going to need a part 3 or 4 to explain this, but it basically, the postal codes are different between species. So you can't just mutate. Okay, so so I earlier showed that we could pump these collagens through a phylogeny program and say, oh, 75% similarity proves common descent. But what they don't mention is if the postal codes are different, that means the postal processing system, if you have a different postal code, let me try to say this. It's like having a different language. each species has their own language, because the postal code here that means endoplasmic reticulum, in another species, it won't be spelled the same way. And so what did they invoke? They invoke things like coevolution, and I call that a major cow pie. I mean, it's just cow pies because you have to evolve.
I've read some of those papers on coevolution, and it's like, oh, okay, and I'm supposed to actually take this serious in terms of what they're trying to account for, and it's very interesting because as you're talking about these additional variables, and
Right.
for anybody who hasn't seen this, we actually had a great conversation the other night about on the prokaryote versus eukaryote dual authentication factor differences that would have to be accounted for. It's a sidebar. Don't go into it in detail right now, Sal, but we'll point some people over to that at some point. The same kind of logic that you're outlining here on the protein level, right? That, hey, we've got so many additional subset variables that aren't optional that have to be accounted for. Is that right?
Oh, there's so much. So like I said, superficially, it'll look like random mutation. And I'm just like, no way, buddy. No way, buddy. It's going to be a disaster. We have a few experiments where you could take one gene in one creature and put it in another and it doesn't die, but that's not generally the case. We have experimental evidence. If we take a topoisomerase from a yeast and put it in a human, it's goodbye, because it has different languages. It has superficially, it's just like I showed the pictures of the cars. So they have the similar architecture, but you're not going to take, say, pistons from one car and necessarily put it in another. There's specific, there's stuff that has to specifically fit. And there are a lot of stuff with proteins here. And I can even, I'm going to go into post-translational modification. Yeah, this is going to be, we're going to have to cut this whole discussion short because.
All right, well, I'll shut up. I want to ask some more questions. You go, you continue on, and we'll get as much as we can. We can schedule a additional one whenever you need to stop.
Follow on.
Go for it.
So now, you can imagine that when we started with, we had kind of like, if you just looked at this, you're like, well, that's not very interesting. It's noise. But what these molecular biologists and structural biologists and biochemists and biophysicists and whoever else getting involved, they're able to find the meaning of these letters. And this is what this diagram is trying to show. This part is the postal code. This part here is the excision region by the procollagen peptidase. That means it's going to target this whole region and just chop it off, but that's very important. So just like when you have a letter and you open it up, so when the letter arrives, you open it up and you can discard the envelope. Well, this is kind of like the envelope here. This is very important to get it addressed, but once it ends up being incorporated into your body, you can dispense with the envelope. And there are other parts here like the NC1 domain. Now, what are these other colors here? Those are what we call post-translational modifications. Those give it, the ones in green give it certain physical properties. These are hydroxylation, and I'm going to go into that a little bit. I'm just trying to show when evolutionary biologists are suggesting it's easy for this to evolve, I just laugh. Because you need proteins. Okay, first off, you need other proteins to be able to process this. Okay, it's not going to work unless you have proteins that can recognize the postal codes. You need other proteins to be able to do excision to kind of remove the envelope. You need other proteins to be able to do these post-translational modifications. Otherwise, you're going to just fall apart because your collagen won't work. Okay, about 20 to 30% of your bodies are collagen. So you'll just be kind of like a jellyfish. I don't know.
And I'm assuming that from the evolutionary timeline, I'm assuming that collagen is considered one of the very early, evolved very early in the evolutionary timeline. Is that a fair assumption?
I think it's only really pronounced in the eukaryotic timeline, and I think it would be in animals and some plants. That's about it. I don't think collagens. I don't know very much. I don't study evolutionary biology. I'm trying not to waste any more time on it. Honestly, this is more interesting. I want to point out something. You see that this is the thing in green is a pea, but you'll notice some of the peas are not turned green. How does the machine know which one to turn green? That's an interesting problem because the location of that pea is really important to making it bend right, because this collagen paralog has to cooperate with other collagens, and it's like kind of connecting, like you have connectors for all the ports on a computer. Everything has to connect right. This is setting up the connections just right. All right, so this is non-trivial. They call that in the collagen world, they call that the chain registry. And this sets up the chain registry. It's not trivial. This is really hard work. People spend a lifetime studying this.
So I'm guessing, even though you kind of mentioned this earlier about, hey, don't just rely on the Douglas Axe 1 and 10 to the 74th functional protein ratio stuff as an across the board, because the numbers vary. I'm assuming that some of the things you're pointing out now are elements that he and other people use in terms of determining what was actually functional, can be considered functional.
Yeah, and they're very shortcuts. I don't know if we have time to cover that. I'll give some shortcuts on how to figure out protein probabilities. Let me just kind of blow through. See, there are also localization signals. Like this is in eukaryotes. You have these localization signals in ribosomal proteins. So when supposedly a prokaryote became a eukaryote, the ribosomal proteins had to go somewhere to the right spot. It had to have nuclear localization signals added. And I'm just like, well, so when the nuclear membrane existed, you suddenly had to reformat all of your ribosomal proteins to have new nuclear localization signals, all new postal codes, right? So this would be like having a city, and you had one set of postal codes, and then what you do is to move the houses physically. You just pull them up out of the ground and move them all different locations in the city, right? You're gonna have to put new postal codes. It's just ridiculous that this could evolve. That's just my opinion. It just seems to me this is where I say evolution needs miracles of special creation to make it work. We showed this, and because we're having some problems with the audio, I was showing how some of the nuclear localization works. So you could look this one, nuclear import and export. Why don't I just show a little bit of it, even if the audio is kind of not working so well?
The nuclear pore complexes are the only channels through which small polar molecules, ions, and macromolecules can travel between the nucleus and the cytoplasm. By controlling this traffic, the nuclear pore complex plays a fundamental role in the physiology of all eukaryotic cells. Most proteins and RNAs are too large to move through the complex by passive diffusion and must move by an active process in which appropriate proteins and RNAs are recognized and selectively transported in a specific direction. Let's consider proteins imported from the cytoplasm to the nucleus. Only a subset of proteins can enter the nucleus, and they are allowed in by virtue of having specific amino acid sequences called nuclear localization signal.
There you go, nuclear localization signal. That is the code: proline, lysine, 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 arginine, lysine, valine. That's the zip code right there.
Hey, by the way, audio is coming through good on this one.
Typically consist of 1 or 2 short segments of basic amino acids. Nuclear localization signals are recognized by nuclear transport receptors called importins, which carry the cargo proteins into the nucleus. With its cargo attached, importin binds to specific nuclear pore proteins in the cytoplasmic filaments. By sequential binding to more interior nuclear pore proteins, the complex is translocated through the nuclear pore. At the nuclear side of the pore, the complex is disrupted by the binding of a protein called RAN to importin. RAN carries a molecule of GTP. This binding changes the conformation of important, which then releases its cargo protein into the nucleus.
Okay, I'm sorry to skip that, just in the interest of time. But isn't that interesting? Look at all the pieces that have to be in place just to get all the postal codes and all the parts going to the right spot. What I often say with these origin of life theories, I said, did you ever, just think about putting a frog in a blender, and what's going to happen? It's not going to reassemble into a frog after you're done with putting it through a blender, because things have to be in the right place. The same with the cell. If you just blow apart all the cell and you just have all the chemicals but in the wrong spot, it's just not going to live. So this whole thing with localization is really important. And one of your favorite topics there, John, the genes that code for the proteins, they have to have little motifs in them so that the microRNAs can also regulate them. So let me show.
Okay, so hang on a second. I want to make a quasi prediction here. Are you gonna, please tell me, you're about to tell me that there are markers inside of the mRNA that are recognition factors for the microRNA to bind? Yes. I knew it.
Yeah, baby. So at the gene level, at the DNA level, we have motifs that serve as regulatory markers, and a lot of the markers are in the untranslated regions. Some are in the mRNA and some are in the untranslated regions here. So this is where John Sanford was saying polyconstraint. So the gene is under constraint to be spelled right. That's why these synonymous changes aren't really synonymous, because it could affect the mRNA, the microRNA interaction with the mRNA. So that's really important. And I'm going to show, so the idea with the microRNA is you can control several different kinds of genes, several different genes, the mRNAs from several different genes. What that means is the gene has to have that motif there. Okay? How do you do that across several genes? And microRNAs are deeply conserved. So that's another constraint on building a protein. The coding gene has to have the correct microRNA targets. And that's why when you're citing those papers on microRNA phylogeny, I said, this is going to be killer. This is just another problem for evolutionary biology. And so these things, you have microRNAs controlling multiple genes, but then you can have multiple microRNAs then controlling multiple genes simultaneously. And so we end up with diagrams like here, where you have these little microRNAs in the diamonds controlling all these genes that are circles. And it's just nuts. This is a regulatory diagram. This is one of the simpler
ones. And then
sometimes,
and in many cases, there's more than one microRNA modifying an mRNA, correct?
Yes, that's exactly what these diagrams are. So you see these little diamonds, HSA MIR15A and MIR503, those are microRNAs. And you can see that it's controlling the TP53. It's regulating it. And it's also simultaneously regulating these other genes. This is a very tough network. The microRNA network is not trivial. And this is one of the more simple ones. And when I was in electrical engineering, we had these are called feedback networks and the connections. They tried to model it. And I'm just like, oh, this is a microRNA diagram. I said, that looks like an electrical engineering feedback control system. So when we have autopilots and all these machines that can control the movement of disk drives, it's the same mathematics. In fact, it's not the same. It's more complex. The God-made feedback control is way beyond anything, because we use linear control. God uses nonlinear control, and we don't really have the math to do that as well as the Lord. So,
well, and just as a quick injection here, the complexity of the microRNAs regulatory piece is just that's an additional regulatory outcome modifier before you even account for things that have happened prior to even the gene expression or the even beginning of the gene expression, in terms of methylation and other variables of that kind that modify it. And what, as a quick interjection, for those who don't know, human genetic engineering that we're doing right now, we're having to create artificial Boolean logic gates in order to control some of the new circuits that we're creating. The irony of that is that from our perspective, oh my goodness, creating a Boolean logic gate, that's some high level stuff. However, the logic gates that are controlling genes that are supposedly derived from evolution, instead of being limited to on or off, have multiple additional variables that can be accounted for inside of that logic gate. And that's before you even account for things that are being shown right now in terms of additional regulatory factors in order to result in the end hyper-specific outcome. And to me, when you really start to contemplate and even understand this on a superficial level, the whole premise of evolution just implodes unless you are going to willfully suspend rational thought. Enough of my tangent. Go ahead, Sal. The floor is yours.
Yeah, you're like Jim Carrey. It's like, you're telling me I've got a chance. So how this relates to evolution, what if you change the microRNA, right? So you talked about microRNA phylogeny. What do you have to do to the other genes to maintain that without blowing them apart? You have to go to each of all the genes that the microRNA regulates and change the regulatory motif, right? So if you change the microRNA that's regulating a certain protein, if you change the microRNA, you have to change the motif in that protein coding gene. That may not seem, that's bad enough for one protein, but look, you have to do that for all the other proteins. This is not, I'm just like, that's why they have to invoke coevolution, and just like, this is nuts, guys. That's a cow pie. Really. That's just, I'm using the word cow pie because you know what I really want to say. But
my favorite is that it's more child friendly, is bovine scatology.
Yeah. So just think that's exactly, see, the problem is if you have a microRNA phylogeny, that they're saying then the microRNAs change, but that also means all the genes have to simultaneously change that it controls. And that's going to, because if you don't, you're going to affect regulatory networks like this. And this is only one of the simpler ones. I've seen just some that are just gargantuan. Don't even, it's beyond human comprehension. It's just unreal. So going back to, so now you're starting to see a little bit of the protein. This is why I want to show the layers of protein probability. And I want to get away from letting abiogenesis guys or evolution guys say, oh, we found a 15 amino acid polypeptide that can catalyze a reaction. It's so easy to make function. I'm just like, well, yeah, it's easy to make a paperweight. You talk about other proteins, it's not so trivial. And collagen is actually relatively quote unquote simple in its function. Its manufacture is not that simple. So we going into post-translational modification. So we have this P here. P for proline. There's a chemical. I'm going to go into some chemical details. This is what proline looks like. It's a simplified version, I think. And anyway, if we add a little modification, the hydroxy group to this proline here, that's what we call a post-translational modification. So that's like chemically, in the chemical sense, we're flipping a bit. So we actually, in biological literature, we actually use the terms like right operation. So you have a right operation. So we have a proline and we do a right operation. And so that's why I had just unenhanced P here, and then I make it green. And that's what you're seeing when I do that, and I add the green there. Those are post-translation. So you had to have a right head go to each individual amino acid that it needs to. It has to go to the right one, which is a non-trivial problem. So it has to navigate to the right one, and it's going to modify it. So that's pretty cool in and of itself. Now you could see some of the other.
Quick question: Is this happening during the protein synthesis, or is this post? Post. Okay, so the polypeptide chain has been formed in the ribosome. It is now left, and then this is, whether it be at the maybe just be chaperones or whatever the process is. These are the kinds of things that are being modified at hyper-specific locations.
Right. So let's walk through this, and I could get the details wrong, and I apologize to the readers. This is worth getting it right. But so you have the first thing is that you have the transcription from the DNA, And now you have a raw RNA. I forgot the term for that, raw RNA. And then the spliceosome comes along and splices out the introns, and now you have an mRNA. And then that goes into the ribosome, and then it makes the protein. The correct term is polypeptide, the raw polypeptide. And it has all of this spelling here. And then it looks, what ends up happening is then the next part is it uses this localization signal and puts it in the endoplasmic reticulum. So we saw that little thing where now the postal code goes there. I may get the order incorrect, but then at some point it's going to strip out this part, basically the envelope, because once it's in the right place, you could just throw away the envelope, right? So the envelope's just there to get it to the right spot. Then at that point, it starts to do these post-translational modifications, putting the P's here. But I should point out, you see these other colors like the K and the S? These are also other post-translational modifications. So you have right heads that do it for the P's, other right heads that do it for the K's, which are lysines, others that do it for the S's, which are serines. These are phosphorylations. These are probably, I had a legend here, which ones these are. It's just nuts. And then you have these C's here. These are disulfide bridge post-translational. This is just nuts. If you don't get all of this right, if you don't have all this, this is just like a factory. So a lot of the textbook biology kind of just, understandably dumbs down the processing of a protein. But for just this collagen, you could see all the post-translational stuff that has the finishing work. So this is when we're manufacturing a car, yeah, we kind of make some of the parts, but there's all this finishing work that needs to be done. Otherwise, it's not going to work. And so these disulfide bridges where they connect the C in the three-dimensional fold, the C has to connect to another C here. This is just nuts. I mean, insanely, it's genius. It's genius. So if it reminds you of the old computer punch cards, yeah, this is information processing, but also at the hardware level. So okay, here are the different kinds of chemical modifications. So the C is a disulfide bond. There are the hydroxyproline. I'm not going to go into there. Yeah, so this little PQ here, this purple, this is where it's going to cut this entire part off. That's post-translational editing. It's just going to dump all those amino acids because this is, again, like opening the envelope and throwing the envelope away. So they're just a ton. I wish someone would make a video of this and you're just like, you'd think, wow. So this is just one protein. By the way, every protein has
question.
I'm sorry?
Quick question for you, Sal. Now, is everything that you're talking about here in terms of the post modifications, are these things all, I'm assuming there are papers on all of these things that you're delineating out now.
Oh yeah.
Okay. So this isn't the reason I'm asking this question is I'm guessing there will be evolutionists and atheists that are watching this. They're going to sit here and claim that this is just creationist bovine scatology and they can ignore all of this. I'm assuming these things that you're delineating out here, the legend you just showed, all that stuff, can be found. If they want to go find it, they can find it in secular literature. Is that a fair?
Right. I'll tell you how to find it. Why don't I just go to Uniprot and type in call1A1, uniprot.org. Type in call1A1 and you'll see all the post-translational modifications. There's a listing. It's easy. Now, what was hard, you won't see a diagram like this because some nut job like me is the one that would actually color this. I did all the coloring myself. That took me a day.
You're visualizing the data. Doesn't mean that the data isn't
right.
recognized.
Why don't I just to I'm going to de-share. I'm going to de-share. I'm going to show how it's done since you asked, because I think it is important that we show how it's done. So I just go to uniprot.org like that, and I type in. I hope you could see this. Oh man, it's not the time for my call 1A. So collagen 1A, and I'm going to click Homo sapiens. So how did I get that coloring? Let me see if I could find it. All right. So when I showed, I'm going to briefly put this back up on the screen. Do you see that diagram with all the colors?
Mm-hmm.
The way I got those colors, this was very painful to do, was Uniprot. I went to all the positions, all the letters have a number associated with it, and they'll say modified residue. So yeah, like the nut job I am, I actually went to position 190 and said, oh, that's going to be a green because that's hydroxyproline. And I went through all of these and put the coloring by hand. And I figured, yeah, I'm going to do it because when you actually see it in a single diagram, it'll tell you this is serious stuff. So like when I had those disulfide bonds, it's right here. So it's in a publicly available website, and you could see it. I took the trouble to make all the coloring. So this can be reconstructed, because I showed this to some colleagues and they said, wow. Even though that they had the data in hand, they worked with it, but when you actually visualize it, that's the wow right there.
Yep. Yeah, it's interesting. I know we need to get you back on track. Sorry for the little diversion there. The to me, the visualization aspect is the barrier to understanding for so many people. Of I think obviously your background professionally enabled you to probably see this in your own head just from knowing what was going on, and then you were able to go ahead and create the visualization. You were able to internally visualize it and connect the dots quickly. But now for the average person being able to see this, it's okay. Takes to me, the reason I love this so much is it really kind of just eviscerates the whole, it's just nothing but chemical reactions argument that we hear so often. But anyway, continue. I know, sorry for distraction there.
Oh no, thank you for the feedback. The reason I did this, do you see, I'm presuming you don't have an organic chemistry background. You didn't have organic chemistry to understand the design. You could see the design if it's just presented. See, how long have you been talking? Maybe 2 hours, and you understand the gist of the problem. It's so clear now.
Yep.
In fact, sometimes getting down into the weeds of the organic chemistry, God bless the organic chemists, people end up not seeing the forest through the trees. And that's one reason it was almost good that I learned the high level stuff first before going back and trying to study the basic chemistry, because a lot of people that deal in computer programming, they understand string processing. If you had to build, just think about trying to build a string processor where you're going to, the job is to identify the P based on the way this works. Okay. In a computer program, you go by specific position, like position 190 or whatever. The way this works in chemicals is they look at the just the letters to the left and right, just a few of them, and how it figures it out, we don't know. It's just amazing because it actually doesn't have to read the entire protein to get the right coordinate. So that's another level of design. So that's collagen. There's different languages of the same post-translational modifications, like for histones, and that's with chromatin. And we're going to talk about 4D nucleome tomorrow. We're going to 4D nucleome to death tomorrow, chromatin and 4D nucleome. And then the topoisomerase has different languages for its post-translational modifications. This diagram I put together by hand also using Microsoft Paint, which was really, really painful. This one was also with, this was done painfully. It took me a day to get this, translating it by hand. So let me play what, now I'm going to get into our topic of the day, which is quaternary structure. Let me play this video of the topoisomerase, because this is one of my favorite proteins since Dr. DeWeese and I study it a lot.
Let's consider what happens as DNA unwinds during replication. As DNA unwinds, it acts like this rope when we pull apart its two strands. As you pull the strands apart, twisting tension builds up in the rest of the coiled portion. It is actually adding one twist to the remaining rope for each twist pulled out of it. At some point, you can't separate the strands anymore. The remaining rope is too tightly twisted. If you relax your tension on the rope, it will twist around itself in a supercoil. It is releasing tension. If you want to keep pulling the rope apart, you have to release the tension periodically, and one way to do this is to cut the rope and splice it back together. This problem has been best characterized in small circular DNAs. There are two methods of dealing with this problem in DNA. One cuts only one strand of the DNA double helix, and the other cuts both strands. Let's look at the first. Topoisomerase I enzymes cut a single strand of the double helix, pass the other strand through the cut, and reseal the break, relaxing the overwound molecule, which now has one fewer twist. Topoisomerase II enzymes do the same thing, but with both strands of the double helix. Topoisomerase II cuts both strands of a double-stranded DNA and passes another double strand through the break and then reseals the break. So if a molecule of DNA is supercoiled, topoisomerase II can remove the supercoiling, 2 twists at a time, to yield a relaxed circle.
Okay, so I just a little bit of to back up. James Downer often says, "Well, you creationists say it all has to be there at once. You know, you're assuming it has to be all there at once." And just like, well, for a lot of things, yeah, because you saw that topoisomerase. If it just goes there and cuts, it's going to shred the genome. If it just cuts and doesn't put it back together, it's going to shred the genome. So yeah, it has to have a way of putting it back together. And hopefully it does some detangling before it puts it back together. And you hope it puts it back together in the right sequence. So I just want to get after James Downer. He keeps saying, you creationists keep insisting it all has to be there at once. I said, well, there are a lot of things that have to be all there at once. Otherwise, it's going to be dead. Get that through your head, Downer. So anyway.
It's hard to get anything through his thick skull.
I'm sorry?
It's very hard to get anything through James Downer's thick skull.
Yeah. So did I have a picture? Oh my goodness. Please tell me I have a picture of what I wanted to show. Okay, I'm going to have to go to the internet. I apologize, John.
No worries, man.
While
you're looking for that, I find that topoisomerase to be very interesting in as Sal was talking about in relation to the what stage. Not only does it have to be in full working order, otherwise you have mass chaos. It also has to exist very, 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 very early in the evolutionary timeline, if not be a co-evolutionary variable that's required. But go ahead. You found it. Go ahead, Sal.
Yes. So this was from my article that I wrote on Creve Info, Creation Evolution Headlines. You'll see this. This is the topoisomerase that we saw the video on, the topoisomerase 2. That's the one Dr. DeWeese and I studied. He's really the one that studied it. He studied this enzyme 15 years. 15 years. He's been studying it. And he's probably going to be studying it the rest of his life. So this is quaternary structure. What quaternary structure, it describes, well, protein complexes that are made of more than one protein. And in this case, we have this topoisomerase. The red is a topoisomerase polypeptide. It's coded from the topoisomerase gene. And the blue is also, it's an identical copy. So to make a working topoisomerase, the cell has to make 2 copies of the same gene to make one. You need 2 copies of this from the same gene to make one functioning topoisomerase. So the left and the right halves are just kind of like scissors. The scissors are symmetric. So there are a lot of things in biology just like in geometry, or you'll see it in a lot of machine parts, there has to be symmetry. There's symmetry makes sense. So in an origin of life scenario, this is where it gets to be really easy to do protein probabilities. I'll say, okay, let's just pick any protein that has quaternary structure. Quaternary structure just means, just to say, it has a structure that needs more than one protein. That may not be the most formal term. Okay, just don't quote me on that as a formal term for quaternary structure, but you're close enough to the truth if you say any protein complex that needs more than 2 copies, it has quaternary structure. There are some that need different kinds of proteins. Those are what we call heteromeric. But for homomeric protein complexes like topoisomerase, homomeric means homo meaning the same. It's like homogeneous. So you have 2 pieces, and so this would be a homodimeric, di meaning 2. So, as I said on after your debate on Sunday, if you had just a random pool of amino acids and you got 1/2, you got 1 copy done. To make it work, to make a functioning topoisomerase, you're going to have to come up with the same recipe if you don't have genes, random amino acid sequence to be in the same proximity to make an identical copy. This is like shuffling cards, dealing them out, and then reshuffling them and dealing out the same sequence. What are the odds? In fact, it's going to be worse because this is about 1,400 amino acids. So that's a very easy way to calculate protein probabilities. That's why I like quaternary structure. You just blow away. It's like, okay, so you need a genome, right, to be able to make proteins of quaternary structure, like topoisomerase or helicase. But, oh, by the way, how are you going to have a genome if you don't have proteins, if you don't have proteins of quaternary structure? You're going to have to invoke other kinds of miracles. So a chicken and egg paradox. That's a very easy way to do protein probabilities. And one thing I'll mention is, you see, the DNA will be kind of like through here. It has to be the right size. So you can't pick any arbitrary size. Just like parts in a car, the number of amino acids affects the size and the shape. And finally, I'm going to show a real quick way, another way to kind of estimate protein probabilities. Kind of like Doug Axe. This is a very crude way, crude with a capital K, all right? But I'll show you, like if I happen to know one protein, it's a beta-lactamase, the one that Doug Axe studied, because I studied the nylonase. Just go to uniprot.org, and since you can play this back on video, this will be a good way to see how you do protein probabilities. So uniprot.org, and I'm going to type in NylB. I just happen to know that's the name of the protein. This is one we find in certain species of bacteria that can digest nylon. And I click on that, and I get all this. It's like a huge encyclopedic description, protein spelled right there. And then I go to, I click the Gen Bank, and I'm going to go to the Gen Bank and click that. And I'm going to go to the gene. And there's a way, when I get to the gene, I can find the protein. When I get to the protein, I click on Identified Conserved Domains. And you'll get this conserved domain thing. And then I can click here, and it'll say bit score. That's the kind of the improbability. Again, this is a crude estimate, crude with a capital K. So if you take 2 raised to the power 130.5, that gives you the improbability. And you could do that with lots of proteins. That's a crude way. This is National Institutes of Health probability scoring. So it's like, well, these aren't creationist scores. These are official scores that they use to distinguish between proteins. They could say that we have identified this architecture, and they could tell you how many bits in probability that it would be by random chance that a sequence could make that architecture. So now we have the tools to actually argue. So I could do that with topoisomerase. I could do it with helicase. So when they say these are just creationist numbers, I'm like, but we use these probability calculations to determine if we're identifying proteins in a genome of a new organism we're studying. We want to know that we're not identifying it just by random chance, or that we're just, throwing our own probability scores. We can recognize like the architecture of topoisomerase or a beta-lactamase. So if I take 2 and raise it to the 130.5 power, that ends up being 10 to the minus 35th power, 39th power, something like that. Now, Doug Axe said a protein of this class would be 10 to the minus 77. So granted, there's a huge difference in the numbers he calculated versus the ones that the NIH calculated, but 10 to the minus 39th is still pretty remote. So,
and that's only for how many for this particular one? How many amino acids are we looking at?
This is 390. Topoisomerase has
so a relatively, that particular one you're looking at is a relatively small protein versus like helicase, which is 1,000, and the the other one was what, 1,400?
1,400, topoisomerase 1,400. So let's kind of do the same for topoisomerase and do a protein probability. Now, the really bad thing for top 2A, the really bad thing for origin of life is, remember, length in humans is 1,531. So granted, humans are pretty advanced, but that's the one I'm more familiar with. Sorry, guys. I don't have one for bacteria, but let's just, for grins. So 1,500, I stand corrected. I was giving the evolutionist, giving them a break there by 100 amino acids. So, yeah, I was.
And just for people who don't pick up on that joke, that 100 amino acids is like 10 to the what, like 20th, something like that, in terms of probability.
So I did a little shortcut there. You can play it back. So let's look at the probabilities here. I'm going to click here on list of domain hits. Oh my goodness. We have bit scores of 1,700. So let me just put that through my calculator. What's the improbability of that? So power of 0.5 raised to the 1,700th power. It went off the charts, John. It's my poor computer couldn't handle it. I'm getting these number errors when I put it in my Excel spreadsheet. So I'll just put 1,000. Okay, this is like 10 to the 301st power, at least. 10 to the 600. It's getting. Let me see. Let me do this. I could do this. Hang on. Let me do 700.
Why are you doing that math? Yeah, this
is
these
like
kinds
10 to
of
the 500th power. 10 to the 500th power. That's for just one. Okay, for quaternary structure, you have to double it, right? Because you need. So what's the probability of that? That would be 10 to like the 1,000th power, and that's for just one round. For goodness, I can only imagine if we did this for helicase. So that's how we can do some of the probabilities. Again, that's crude with a capital K. So I interrupted.
Right, but the irony of when you're talking about it being crude is that there's actually additional variables you could insert into the probability that would not make it go down. It would only make it go up. Is that a fair assumption?
Yeah, it'll be even more improbable. Yeah.
Right. So like if anybody like wants to talk trash about the that's just general, you're not really accounting. It's like, yeah, you're right. I'm not accounting for everything. The only if you want to argue with whether or not my position that's being taken is a fair one, well, let's go do the math for real, and you will end up in a worse position.
Yeah,
I mean that's kind of where I look. That's the way I look at it anyway.
Well, I suppose people say, well, there's still a remote probability it could happen. It's like Jim Carrey and Dumb and Dumber. You're telling me I got a chance.
Well, and that's something that really I just blows my mind at the way the evolutionary advocates double down on this. And it's like, okay, fine. You want to rely on deep time. Congratulations. Your own stupid evolutionary timeline gives you no more than 500 million years, and in some aspects, as low as 200 million years, in order for things like this to have occurred in an undirected process. So time is not in your favor, especially from an abiogenesis, from abiogenesis or a traditional evolutionary perspective. It's not in your favor at all, and it only gets more and more out to lunch, in my opinion. The further and the more you dive into these kinds of things.
You know, I just realized this is only 90 minutes so far. We covered a lot in just 90 minutes. Amazing.
Now this may be a good spot for us to stop. Do you want to do we'll plan on doing another round to go into the next layers in more detail?
Yeah.
I know there's additional things you were wanting to cover, but
yeah, I'll have to think about it. But I mean, I think we covered a lot of really good ground today. I really enjoyed. I got a lot off my chest because there's a lot that I've heard on the internet about origin of life and protein probabilities, and it's like a lot of times we are just quoting, and understandably so, we're quoting other people's research. But what I tried to do today is in 90 minutes, I tried to show how you can get a sense for the probabilities just from scratch. So when I showed the primary structure and then the secondary structure with the alpha helices, that shows exactly why we need the homochirality. That's one level of probability right there. I mean, if you have 1,500 amino acids, this is like getting it heads 1,500 times. That's the first level. That's not even including the sequencing with the correct amino acids. You just have to add them all left-handed. That's just the start. I mean, there are just so many levels of this. And then I was showing it was great to show like the parts of the protein with the postal codes, what we call the localization signals, and then all the specialized post-translational modifications and all the proteins that would have to be involved. And then not to mention at the gene level, the microRNAs controlling the multiple microRNAs controlling the regulation of the gene before it even gets all that processing. It's just, I'm surprised we were able to cover it in 90 minutes. And
well, you know, one of the things that really, as you're showcasing and explaining more of the additional layers of complexity that we're actually having to be accounted for, the importance, in my opinion, of things as simple as like peptide versus non-peptide bonding, right? And how that applies in the relation to these things forming by chance, you know, and just the implausibility, for me anyway, of, hey, we've removed all of the control variables from the equation in terms of how modern life is able to make all these things come together, right? We have to, for abiogenesis, we have to remove all those things from the equation. And you're dealing with pure, hey, they got a bond in the correct order at the right time. And it's got to be these, and there's no way around it. And the whole aspect of, I think, is also lost in a lot of people of the, flipping the coin 1,000 times, it's all landing on heads. It's like, yeah, but you guys are forgetting that not only do they have to all land on heads, you have 20 different coins you have to flip. And it has to be the correct coin that also lands on heads every single time. Yeah. And when you start inserting and there's more, and for those who don't know, there's multiple additional variables that I'm not even talking about yet in terms of this, how does all this come into play? And I think those things just get completely glossed over in the abiogenesis rhetoric, and then, you know, the loud screaming from the mountaintops that we hear from a lot of the online activists about how this is just solved, and all the way to certain people who I will not necessarily name names at the current moment who happen to have PhDs who talk about how these things are, basically solved.
Yeah.
And it's like, no, they are not solved. And while you may be claiming they're solved, you go and read the papers that are written by, synthetic chemists and organic chemists, high level, and they're like, wait, what? No, you didn't account for this, and you didn't account for this, and you didn't account for this, and you didn't account for this. What are you talking about? And anyway, not to go off on a diatribe there, but those things just really, they both irritate me and fascinate me in relation to the level of blunt denial that huge portions of academia are living in, to not see the obvious. And you mentioned it earlier, they not seeing the forest for the trees.
Yep. And I thank God for people like James Tour calling a spade a spade. Origin of life research is retarded. I mean, that's pretty cruel thing to say, but it's like, yeah, I'm glad someone of his stature is really calling it for what it is. And
for those listening, if you've never read this, you should go. I'll post a link to it in the description. There's a great, very, very detailed article. That article is longer than most research papers, where he did a direct breakdown of what is actually being argued and doing direct comparisons to extraordinarily simplistic synthetic chemistry activities that they're doing. And if you read what they had to do to pull off these comparatively simple outcomes, and then you look at what is being executed in some of the simplest proteins, you will, to me, when I read through that, I'm like, man, if you actually look at these comparisons and conclude that this stuff is possible without an intelligent agent, the supreme intelligent agent in this context, being required, you have suspended all rational thought. I can't really find any other way around it.
Right. And one thing, as you could probably guess from my presentation, this is what Dr. Sanford and I talked about, is when we make it a little bit more visual, you can actually see the improbabilities. Something in the mind, it starts to click. Because when I started to, I saw it in my mind, I said, if I only could make this on a picture, you could see the difficulty. And so that's why I really like structural biology. So it's been a real privilege. And I want to thank all the faithful listeners and viewers who've supported this channel and have been watching. And hopefully you've enjoyed our presentation. It's been a real privilege to have an audience to be able to share this with. So I want to thank, I haven't been able to look at every comment, but I really want to thank everyone that's been supporting our channel, and even the critics, because they kind of make things lively. So
well, it's always fun to be able to read their denial as you are debunking the foundation of their positions, and they still continue to cling to it and claim that it's, liar, liar, pants on fire, you're making this up. Look at that Christian fantasies and all those different common taglines that we see. It's like, yeah, sure, okay. Using the data, everybody's got the same data. Yeah, just look at it. But okay, so here's a question I've got for you, and I'm guessing from the some of the people who have been watching this, there's gonna be a few who are gonna be probably showing up for your debate against Dr. Dan Stern Cardinal tomorrow. Are you gonna be bringing some of this heat in your debate tomorrow?
Unfortunately, no. This is the fun stuff. Tomorrow we're going to talk about evolutionary biology, which is just like so boring. I won't be able to talk as much structural biology and chemistry as I'd like. So if someone wants to get the tenor of tomorrow's debate, there are a couple of videos here on Logical Plausible Probables channels, one on Mueller's limit and the other on Fisher's theorem and natural selection and fitness. That's more, and those are things that I try to do well at, but it's not what I really love. So you're seeing a little bit more. I like the chemistry and the physics side. So the stuff I really like, though, I don't really get to do, which is physics, because right now all the easy stuff is in biology, meaning the others, it's we're getting creationism evidence for it handed to us on a silver platter by the researchers at the NIH and biotechnology. And it's just right now, it's just like a candy store. You just Google on ERVs and function or transposons, and you can't really say ERV and function. You have to say things like ERVs and architectural proteins or transposons architectural. You have to use the right words.
I've seen some
And it's
stuff
like a
also with
candy
like
store out there. Just every
ERVs. I've also seen if you toss in like regulatory factors in associated with them too. I've seen, or gene regulation. I've seen some papers on that as well. So you said ERVs and architecture. So you said on one of them.
Okay, so
and then,
how did I find it? Chromatin organization ERVs, or chromatin organizations and transposons.
okay.
And some of the papers I'm finding are just blowing my mind. John, it's just like it's a candy store out there. And John Stanford said, we need to have a website. We're just going to be giving away candy because there's so much of it. So I'm building a new, I have all these, I have the evidenceandreasons.org website. He said, well, just have one for science where all you're doing is putting all the wow in biology. And I own the domain name, I think. I'll have to look. And I'm going to roll that out where we're just going to have all these wow factors. Because right now, biology, as far as creationism is concerned, is just like a candy store. And we have all these quotes from people like Francisco Ayala, who was a Catholic priest, turned evolutionary biologist, and then got fired for sexual harassment. Sorry, I just had to mention that. He was saying, look at all those transposons, that's junk. And then John Avise was saying, yeah, 50 to 70% are repetitive elements. That's all junk. That's all, well, but that's only junk in their minds. What's being junked are their ideas, because we're seeing that those repetitive elements are really important for 4D nucleome organization. The 4D nucleome organization affects things like, if that organization is broken, it creates cancer. That's why it's being studied. Or all these other cellular problems. So 4D nucleome, we're going to hammer that. I'm going to hammer that tomorrow and say that junk hypothesis is, the only people who maintain it are evolutionary biologists and stupid horror energy. Really.
Well, it really is. It's sad almost. It's becoming very sad, in my opinion. I'm being able to, from a personal level, interactions with you and then my own searches for different papers. For me, a lot of my searches are derived from, okay, I kind of approach, and I wager you do to an extent as well, but I kind of operate from the perspective of, okay, assuming this is designed, what would make logical sense as a requirement, right? And then I will go and try different search queries in relation to different components of biology. And to your point, kid in a candy store, like, it's extraordinarily probable there's going to be an article, a paper, or a review article, or whatever, that is directly addressing whatever this random thought. And what cracks me up about that more than anything, just from a personal perspective, is I am not a biologist, yet I am being able to deduce what should be there, and then go and read their research, and which is why I find it interesting in context of the pushback we hear from the atheists and the evolutionists of models and predictions and all that kind of stuff. I'm like, okay, cool. So I'm being able to predict something that's going to be discovered relatively recently, without being a biologist and without being privy to all the other underlying components. I can come up with an idea, Google it, and boom, there's a paper. And you're telling me that I can't make a prediction based on pure logic from the perspective of, it must be designed, therefore there must be structure, therefore what is a reasonable conclusion of a prerequisite requirement? And then presto, there it is. And I'm sure you've seen that exponentially more than I have in all the last 6 years of all your research. But
actually, you and I are kind of on the same lines, because that's how I did my Googling. Is I used kind of the engineering instincts. That's why I got hired by John Sanford, even though I wasn't a biologist. He said, he felt I had the right skill sets, which was engineering and physics, oddly enough, gambling math. uh, 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 so you're right, and this is going to hurt. I mean, I'm not trying to. Well, maybe I'm going to make some derision about evolutionary biology. You know, the people that are making all the breakthroughs are chemists and engineers and computational types. The people that we have on the research teams, the last person you need is an evolutionary biologist because they don't have the right skill set or the mindset to understand. I mean, how helpful is it if you're studying the 4D nucleome and you're studying repetitive elements and you come with the mindset, this is junk, and they're trying to solve problems in cancer, and they know 90% of all of these disease indications are associated with these with the so-called junk DNA, which would be half of that would be the repetitive elements. That sort of mindset is unhelpful to making medical breakthroughs. So that's why someone like Joe DeWeese, who's a young Earth creationist, who's doing cancer research, he is more valuable to the medical industry in that field and what he does. They would sooner hire him to do this research than they would an evolutionary biologist because he has the right skill set. Same with engineers at MIT. And this was a study that was done some time ago. 1/3 of the biologists, 1/3 of the engineers there at MIT were studying biological problems because they had the right skill set. They had the right skill set. You need engineers to do this. Because these are, why? Because these are machines. I just had to say that again. There's someone I know that every time we say that, it gets under his skin. Machines.
Well, and that's what I just.
You want to understand machines, you need mechanics, you need engineers. You don't need evolutionary biologists telling you it's junk and it's random. That's the wrong skill set. That's the wrong mindset. It's unhelpful.
Well, it's interesting as you're kind of breaking that down, but also showing the real world application of skill set and worldview almost. Now, I'm not suggesting that all the people that are the engineers and whatnot don't necessarily believe in evolution. That's not the point I'm trying to make. But the way their mind operates when they're addressing a problem is a much different one than, oh, I'm going to try and come up with a theory of how this could come into existence without intelligent agents or without a structural process behind it. And as you and I have talked about before, for me, from the information technology side, to me, it was obvious. Like, as soon as I started learning more about it, oh, well, that makes sense. And then since then, I've learned much more about the structural components and the engineering side of it in terms of how the information component is executed. And it's like, oh, okay, cool. This is the physical manifestation of prescriptive programming. Like, there's no way around it. Like, it's just physical. That's all this is. It's why I find it so fascinating, because you've made the point many times about the lines blurring between hardware and software, and which I agree with completely. And I've made the point of many times of, so you're suggesting that because an old, I say old, starting to be old, magnetic hard drive is being replaced with solid state, right? But you're suggesting that because the magnetic hard drive used magnetism to store data and express it and have the read-write operations, all that kind of stuff, you're arguing that because that used physical process, that the human brain wasn't required? Is that what you're saying? Because it's the literal comparison, right?
Yeah.
And I think everybody tries to delineate and separate between the hardware and software without accounting for the source origin requirement. I'm talking about in the evolutionary biology world. And that's why I find it so interesting in papers that I read where they acknowledge these things. They acknowledge the machines. They acknowledge the coding. They acknowledge the process, the information processing, all these different pieces. They acknowledge that they exist. And then immediately in the conclusion, talk about how it must have evolved. I find it mind-blowing of how they use all the phraseology of engineering, information technology, all those different pieces throughout the entire paper, and then conclude with, it must have evolved. I'm paraphrasing, obviously, but I'm sure you've read that in an innumerable number of papers. However, I have started to notice that there are some, fewer than used to be, or more than it used to be, that you're not even seeing the word evolution in the entire paper. Have you noticed that?
That's true. I think there are probably some old school editors who say you didn't mention evolution and kind of force the writers to put it in as it's kind of a token gratuitous acknowledgement. It just so but yeah, I read some of these structural biology and biochemistry papers. They don't even mention it. I said that's so beautiful. There was one paper that talked about the 4D nucleome being a three-dimensional data storage system, and I knew exactly where they're going. They mentioned evolution once. They mentioned structure 71 times, and just like, yeah, structure is more important. And a lot of, just like what you said. Before I go and thank you again for this time, I wanted to say 20 years ago, and this relates to tomorrow's debate, I actually thought most of the DNA could be junk. I mean, I didn't know any better. And having been around 20 years ago looking at this debate, there wasn't anything that could overturn that. I mean, the evolutionary biologist made a very compelling case. But it's amazing, just like in a murder trial, if a detective keeps finding evidence to kind of acquit, the defendant, it gets exciting. It's like, oh my goodness, the case looks so bad and it's changed. That's what's happened in the last 20 years. And God he sent people to help the creationists when the creationists couldn't help themselves. And those people are the people in the medical and biotech industry, because they're getting billions, tens of billions of dollars to find all this complexity out. And, there's no way creation is like the $40 billion a year budget at the NIH. But we have a God that cares about people having their faith built. And we're privileged to live in the 20th century where we could see that we're fearfully and wonderfully made, just as it says in the Psalms. There is some junk in our DNA because of genetic entropy. So the difference between intelligent design and creationism, and in intelligent design, we say biology is designed. In creationism, we say biology is designed, but it's also cursed. And we see that on many levels. So there is some junk, but God has, I feel God has ordained this time, just like when he had Pharaoh just look so powerful for 400 years, and the Pharaohs were oppressing the Israelites. And then Moses came along, and, just all that buildup where the guy was just dominating kind of made it more sweet when the Israelites got liberated. And this is what's happening now. I feel that the evolutionists have had such a stranglehold on science, and now they're just being set aside. I was at the ENCODE 2015 conference after Dan Grauer saying all those nasty things that these people were ignoramuses and crooks, just some nasty things to say. And that's like, he's treating them like he treats creationists. But I was at that conference. There was not one mention of him. He's like nothing. He's irrelevant. And I'm just like, yeah, that's what's happening. We're breaking free of the stranglehold that evolutionary biologists have forced upon us to say that we're random accidents and we're junk. Anyway, I got to go. God bless.
Thanks so much, Sal. Appreciate you as always. And everybody who's stuck around for all of this and for this great discussion I've had with Sal today. Thanks so much, Sal, for your time, sharing all this knowledge. I'm looking forward to learning more, and I'm very much excited about the debate tomorrow with Dr. Dan. That should be a good one. And, as I always love to say before we wrap up, actually, I'm going to use a little personal analogy in context of what Sal was talking about today. I love to play poker, and I feel like the 56 since the discovery of DNA, we've been, that was the kind of the beginning of the hand, if you will. And there's the flop and a round of betting from the evolutionists and the people who believed in intelligent design, and then call. And then there came the turn in the 80s, 90s, and the evolutionists doubled down as the Human Genome Project came forth, And then now we're getting into the understanding of the structural biology, the interdependent complexities, and how so many of their foundational pieces of the puzzle and phylogeny and other aspects are not anywhere near as sound. And then comes the turn, the river of this hand, and the evolutionists have gone all in. They pushed their entire stack in the middle and said, "I dare you to call." But as we've been doing analysis, as we learn and we read and we pay attention to the discoveries that are being made, well, guess what? We've seen the tell in your bluff. We know that you're full of it, that you're not actually as confident in your position as you once were. You're starting to sweat. That little drops of sweat are starting to roll down, and we see that, we identify, and we see that that carotid artery in your neck is going really quick. So guess what? Those of us on the intelligent agent, intelligent design, the creationist side, we call your bluff because your entire position is flawed, is so many assumptions that defy logic. And as the more and more people begin to recognize and realize, and the information that we're being discussed on this channel and many others comes to the forefront and is being recognized by the average individual, you can no longer play your games of just, oh, well, people don't understand this. Oh, it's too complex. Because you know what? We can understand it. It can be explained. And in the not too distant future, you will recognize that your bluff has been called. And those like me are going to say, ship it. Shipping those chips, baby. We win. You're going to be stacked. And you're going to be walking away from the table realizing you shouldn't have gone all in on that moment because you had nothing. You had 7 deuce, and you're up against the nuts. Anyway, that's enough for me today, ladies and gentlemen. I hope you enjoyed. Stay tuned for more stuff that's happening this week. And as you consider the different things that are discussed here and in debates and all over the internet and your own interactions with the atheists and the evolutionists in the chat and the comments, always remember the conclusions that you reach must always be logical, plausible, and probable. Thanks so much, y'all.